Today, it's an honor for us to have with us one of the experts in stem cell in Southern California. She's probably the recognized authority in this field, Dr. Aileen Anderson. She's a UCI professor of physical medicine and the director of the Stem Cell Research Center at UCI. She did her bachelor's degree at the University of Illinois in bioengineering. She did her PhD at UCI in biology and microbiology. She did a fellowship at Harvard University in neuroscience. She has a very special interest in spinal cord injury, for which stem cells are used, and for glioblastoma, which is a serious malignant disease of the brain. She's published over 90 studies in the very best professional journals, including neuroscience and neurology. And she's going to be talking to us today about stem cell research. And so please welcome Dr. Aileen Anderson.
Thank you so much. Good morning, everyone. I'm really pleased to see you all here because I know times are a little bit uncertain right now. UCI is certainly grappling with how to respond to the spread of the coronavirus. So I'm very glad that you all took the time to come out because I think this is a really important topic that we're talking about. I'm going to try and give you a very broad overview, really about two things, about stem cells, what they are, what our center is, and sort of where things are, but also stem cells as they're out in the community and where we are going forward in 2020, because in California, this is certainly going to be a very pressing issue over the next 6 months. Is that loud enough? All right. I am very happy to make this as much of a discussion as you would like. We don't have to be super formal. So if I'm giving a slide and you have a question about it right then, please feel free to raise your hand or give me a nod and ask a question as we go. I'm at your disposal this morning. So I thought we should just spend a moment and introduce about who I am and where I'm from a little bit more at the Stem Cell Research Center at UCI. The Stem Cell Research Center here is over 50 faculty from across 6 schools and 21 departments on campus. And so it's an extremely interdisciplinary center. We span College of Health Sciences, the School of Medicine, the School of Engineering, even arts and the School of Law, because we have a component that's about ethics in stem cell research. Our mission is really threefold, as you can see here. It's about basic science discovery, understanding stem cells and how they can be used. To teach, we teach undergraduate students, graduate students, medical students, the whole kit and caboodle, and to heal, to drive what we've learned about stem cells and stem cell biology over the last 20 years or so forward into clinical trials. And actually, UCI has been a real player in that side of things. So starting out with stem cells and just the origins of stem cell research, for those of you who are interested on that side. You may remember, going back not that very long ago, that stem cells invoke some really controversial and disparate feelings among people, both within the sciences and within the community. This is a great slide, I think, because on one side, it's a demonstration against stem cell research, which some people feel very passionately about. On the other side, a demonstration in favor of stem cell research, that stem cell research is not being given the resources to move fast enough and far enough. These two protests took place on exactly the same day. So that kind of encapsulates where things are in a nutshell, right? These very disparate ideas. So what's the sort of history to that? What are stem cells? These are undifferentiated cells. So a cell that's not committed, that's not specified, and they divide by a process that's called mitosis. In fact, if we look at the screen up here, the 2 cells you can see that are connected by a line of green, that's a stem cell that's in the process of dividing as we look at it, as we caught it in the microscope. The 2 cells that come from one cell are called daughter cells, and they have a choice. They do one of 2 things. They can either differentiate, that means they become specified, they decide what to be, or they stay as a stem cell. So they propagate in a process we call self-renewal, an ongoing pool of stem cells. Why is that important? You have stem cells in your body right now. All of our organs and tissues do. If you have a cut on your hand, you require stem cells for wound healing to heal that cut. It's very important that we maintain that pool of stem cells throughout our lifetimes, because when that pool starts to fail, in fact, our capacity of our bodies to heal ourselves starts to decline. Okay, so an important thing to realize in that context then is stem cells make up all of our organs and tissues. That's true during development, and it's true in the process of self-renewal that we just talked about. So if we look at this first picture that I'm showing you, that's a tree. That's to represent what stem cells are in your body. At the base of the tree are cells that can literally make anything. And as we travel up the branches of the tree, these are cells that become more and more differentiated, or what we call specified. So we can break that down into a couple of terms that it's important to understand. These cells at the base of the tree that can make anything are the cells that are pluripotent, pluri meaning many. And this is a type of cell that's an embryonic stem cell, about which we had a lot of conversations in the early 2000s in the United States and beyond. And they derive from the inner cell mass of a blastocyst. That's their source of isolation. If we go farther up that tree into the area where we have multiple branches, these cells can make different lineages that compose all of our organs and tissues, but they can't make absolutely anything. So for example, if we go up the ectoderm branch, this is what I study, the central nervous system, and these cells can contribute to brain and spinal cord. They can make the three kinds of cells we find there, neurons, oligodendrocytes, and astrocytes. They're still a stem cell. But they can't jump over to a different branch and make mesoderm and endoderm. They can't contribute to lung, for example. So they're specified partway, but they can still self-renew, and they can still make multiple cell types. So we call them multipotent stem cells. Make sense? Okay. So multipotent cells are very important in that regard. And I want to give you now just a little bit of the history of why we started thinking about stem cells to begin with. And this started actually with a very fundamental question about biology, discovery biology in the 60s. So in the 50s, we had isolated the structure of DNA. We started to understand DNA, that there was a sequence that made up our genetic code, and this determined fundamentally who we are. But we actually didn't know whether fully differentiated, these specified cells up here, adult cells that are in our organs and tissues, were genetically identical to the cells that were present during development. We didn't even call them stem cells at that stage. So in other words, when you had a fertilized egg, were those cells, was the DNA the same as the DNA that ended up out in these differentiated bits? So that may seem obvious now. It wasn't at all obvious at the time. And so this man, Sir John Gurdon, who won the 2012 Nobel Prize, I'll come back to that for this work, asked that question. And he asked it in amphibians. So these are frog eggs. And what he did is he took the fertilized egg, you can see down here, imagine at the bottom of the tree, of a frog, and he replaced the nucleus with cells that had come from a differentiated cell type up at the tip of the branches. And his reasoning was, if they're genetically identical, when I make that swap, you should get a normal frog. And in fact, you do. And so he answered that fundamental biology question in a way that really shaped how stem cell research went forward. If we jump forward 35 years to 1997, this gentleman, Sir Ian Wilmot, did the same thing for the first time in a mammalian species. It took that long, 35 years. Not that people weren't trying. And what he created, you may remember at the time, a buzz in the news, was Dolly the sheep. And that was the first big success of that process called somatic cell nuclear transfer that became the basis of our understanding of stem cells. So what after Dolly the sheep then? We had answered this question, are these two types identical? Yes, they are, genetically. Interesting, what we found out of this is they're still not really the same. And this harkens back to really what is the most exciting area, I would say, of probably biology and certainly stem cell biology right now, is our very leading edge is understanding how the program is different, even though the DNA is the same. And that's a process that's called epigenetics. And I'm not going to talk about it today, but it is the leading wave, probably not just of stem cell biology, but of biology overall. So we have this. We have these pluripotent cells, as we talked about, at the base of the tree. Pluri being many. Again, this is the definition of embryonic stem cells. And these two findings really set us up to move forward in terms of understanding stem cells and stem cell research. But in 1998, along came this guy, Jamie Thompson. And what he did was to crack this code for how to isolate these cells that are within the fertilized egg and get them to grow in a dish, mammalian cells. And he did that in particular, not just for cows, which is where he started, but in 1998 for human cells. And thus was born the human embryonic stem cell and all of the for and against controversies that I highlighted in that first slide. Important to understand, though, and so I'm going to say just a couple of words about embryonic stem cell policy and how this policy became functionally irrelevant pretty quickly. So what do I mean by that? The start of embryonic stem cell policy, so Jamie Thompson made that discovery in 1998. By 2001, it was a buzz of controversy, and George Bush made the decision, with a lot of input from all directions, to limit federal funding for human embryonic stem cell research. This functionally banned embryonic stem cell research across the United States. In 2004, in response to that, not shown up here, came the California Institute for Regenerative Medicine. And we'll come back and talk quite a lot about CIRM and the California Institute for Regenerative Medicine, which was passed by a ballot initiative in 2004, where an overwhelming majority of Californians voted to say we should support stem cell research regardless of the federal ban that was placed. So that moved forward 8 years to when President Obama was in office. He lifted those Bush restrictions, and embryonic stem cell research, and in fact other stem cell research, became possible across the country and not just within California. By November of last year, in fact, there are over 410 embryonic stem cell lines approved within the United States, and more if we count those that have been generated from outside the United States. I should just mention, and I'll come back and touch on it, that approval process is an extremely rigorous one from an ethical point of view. But what do I mean up here by how this became irrelevant? I mean this. In between these periods, in 2006, this gentleman, Dr. Shinya Yamanaka, developed a new understanding about stem cells, and that's the concept of induced pluripotent stem cells, being able to make a cell that behaved like an embryonic stem cell but didn't derive from an embryo. And for this, he shared that 2012 Nobel Prize with Sir John Gurdon. So what is an induced pluripotent stem cell? The idea here is that we take a cell that's from very far up these branches and do what's called reprogram it. So Yamanaka discovered that you could use just 4 genes, 4 factors, and teach a cell from out along these branches that is fully differentiated, fully specified, to behave like a cell that was down at the trunk of this tree. And that's what a reprogrammed or an induced pluripotent stem cell is. So these are the 4 factors we talk about, and that opens up a world of difference, a sea change in terms of how we can think about stem cells. So embryonic stem cells, we thought always about differentiating them, specifying them to behave like a cell or tissue that we might want to replace, for example, dopamine neurons in Parkinson's disease. But we always have a problem with that process for whether or not the person we give them to is going to reject them, right? And we can't study in a very patient-specific way. What if one person has a disease that's not functionally behaving like someone else's disease because of our genetics, because of our code? It's different. These cells open up a path to be able to get at both of those things. So once we teach them to be a pluripotent cell, now we can think about if there's a genetic defect, we can correct it in those cells and give the person's own cells back to them, right? We can think about taking these and differentiating them partway, bringing them up this tree, but studying in a person-specific, disease-specific way mechanisms in a dish by growing those cells. Screening for new drugs, right? Not everyone in reality responds equally well to the same drugs. If you're on a drug for high cholesterol, everyone doesn't have the same response. This is why we tend to switch medications and try several before you settle on one. That gives us a potential to do patient-specific disease screening in a dish. By the same token, we can now think about if stem cell therapies are a good possibility for people moving forward with some diseases, which they are. We can generate patient-specific cells and use those in our clinical pipeline to transplant back, right? So the capacity to generate induced pluripotent stem cells changes the game. And again, as I said, kind of makes functionally irrelevant many of our earlier controversies about stem cell research. So with that, I want to transition a little bit in terms of background and talk about this area of hope versus hype, about the idea of CIRM and the California Institute for Regenerative Medicine, and about some of the things that are going on across this state in terms of stem cell research. And I'm going to highlight in particular the Alpha Stem Cell Clinics and the Alpha Stem Cell Clinic Network, because this is something that is unique to California and an area where we've really, in the last 14 years since the original CIRM funding, 16 years, I guess at this point, made some surprising leaps forward that we wouldn't have anticipated. All right, so let's jump forward into this next bit, because I think there'll be a lot of interest there as well, and I want to make sure we have time to talk to you about these pieces. This is just a cartoon, in case you can't see it, that is, maybe looking forward a little bit. This gentleman has brain cells in his pump, and he's filling this guy up. But the point I want to make is that stem cells, and the point I want to make in this section, really will revolutionize the treatment of human disease. And in fact, we are far along that path already, which I think is really exciting in terms of the progress that we've been able to make. But it's important to spend a couple of minutes and talk about the ethical aspects of the clinical use of stem cells. And there are some points here, I think, that are important to understand. Stem cells, as we just talked about in our back and forth conversation, there are a lot of things that we don't know about them or fully understand yet. And so there's always a risk-benefit ratio that we have to think about in medicine and very carefully consider. And stem cells, I hope you have the sense from that conversation, definitely meet the definition of a complex situation, right, at all different levels. It may be that in some cases, cell therapeutics are better than the current standard of care. It may be in cases you're better off taking a drug than you are taking a stem cell, or having a surgery than taking a stem cell. And stem cells are at early days. We're learning to understand that balance right now and really where it is. It could be that stem cells are worth it in chronic conditions and not acute in some cases. And maybe that a stem cell would be the best therapy in a mild case versus a severe case, or the opposite, depending upon what the situation is. So this balance is really an important part of where we are with stem cell research and understanding that balance right now. On the other hand, there are certain rare conditions, or in some cases not so rare. Traumatic brain injury is a great example, where we have no real treatments. Stem cells may be the only option in terms of moving forward for some complex diseases. And so again, that comes on our risk-benefit scale. But both of these things introduce really complex issues of making sure that the people that are getting those potential therapies and enrolling in clinical trials understands that risk-benefit ratio and all of the pieces that go with stem cell research. And so that's one of the challenges that we have as a field. Another set of challenges involve how do we do equitable distribution of these cell therapeutics, just like some of the new generation cancer therapies that are coming online. In fact, stem cell treatments are likely to be very expensive. We have to grapple with how are we going to roll that out into the clinic so that only a select few aren't able to get them. In fact, CAR T therapies for cancer, which some of you may have heard of, this is an example of something that really is dependent on stem cell research. And they're wildly expensive. These can be $1 million a therapy, right? Should that be acceptable in terms of how we set up our clinical translation, thinking about where stem cell research is going to be in 10 years? And the reality that, as we highlighted at the very beginning, there are some people that just don't agree with stem cell research, and we have to be respectful of that. We have to encompass that in terms of what our long-term planning is. So I would, some of the things I'm going to give you here are just resources that you might want to think about. This was a great article in the New Yorker that appeared at the end of last summer, Promise and Price of Cellular Therapies. It's easily Googleable if that would be something you might be interested in reading more about. It really specifically addresses the idea of price and how to do equitable pricing and distribution into the community. Another aspect of the ethical research of stem cells for clinical use of stem cells we have to grapple with is how do we manage enrollment in clinical trials? And I want to highlight this right now because I'm going to start talking about the difference between clinical trials with stem cell therapies and direct-to-consumer marketing of stem cell therapies. And so I want to make some of those distinctions for you right now. So bear with me a second here. Really what I want to point out is that in a proper clinical trial, these are highly regulated processes, regulated by the Food and Drug Administration. All of the preclinical data that supports a particular cell therapy for a particular disease indication has to be vetted by the Food and Drug Administration. It gets looked at very closely. With their authorization, you can move forward to a trial. Any research, whether it's clinical or basic science at any university around the country, gets reviewed by what's called a Human Stem Cell Research Oversight Committee that's very closely vetting of where those cells came from, were all the appropriate ethical approvals obtained from the people who had donated or consented to be involved in a process. And of course, any human subjects work that happens at an academic institution is reviewed by what's called an institutional review board or an IRB. And that process is, again, critical in terms of making sure that people that are involved in this research understand what they're getting involved in, and that the cells that we use, whether it's at the bench or at the bedside, have been derived and brought forward in an ethically responsible manner, right? On the other hand, in terms of moving responsibly towards new therapeutics, we have a couple of competing ventures. One is just hype, the amount of hype that is out there about stem cells. I'm going to show you a couple of slides of that in a moment. Something that we're very concerned about, most people who do stem cell research, certainly us at the center at UCI, which is direct-to-consumer marketing, and I want to talk to you about that a little bit. And because of direct-to-consumer marketing, an increasing number of adverse events that are coming out. An adverse event is when you receive a therapeutic, whether it's a drug or a stem cell, and have a bad reaction to it for some reason. And again, I'm going to give you a couple of examples of that. So when I started doing stem cell research in my lab in about 2002, it was very early in the days of stem cell research. By the mid-2000s, end of 2000s, 2008, there had been just a proliferation of clinics across the globe. All of these countries that you can see highlighted in blue are countries where you could leave the United States and go to get a cell therapy for something. I work in the area of spinal cord injury. People were traveling to Russia, to Portugal, to the Dominican Republic to get injections of God knows what, on the slim chance that it was going to make them better, and not thinking about that risk-balance ratio that we talked about. So we started out with a lot of concerns for stem cell tourism, those of us who work in the field, but we were thinking very much from the perspective of people that we knew, patients that we had interacted with who were going overseas for these things. The reality is that this has changed significantly, and now we have stem cell tourism, but it's happening in the USA all the time, and it is poorly regulated and not well understood, I think, by the consumers that are going out to access this. So we call this direct-to-consumer marketing. This is from a review paper by Lee Turner and Paul Knopfler that was published in 2016. In fact, UCI just recruited Lee Turner from University of Minnesota. He's going to be our resident ethicist in terms of ethics and policy at the Stem Cell Research Center. Every single one of these red lunks, red dots here, is a stem cell clinic that was conducting direct-to-consumer marketing at the time that they put this paper together. We know that there's only more of them now. The number hasn't gone down. Very importantly, California, Colorado, and Florida are the really hotbeds for this. California ranges between being ranked #1 and #3. I think we're currently #3 in terms of the number of direct-to-consumer marketing clinics we have. So This is an example of stem cell hype. And stem cell hype comes out of direct-to-consumer marketing clinics. I put this one up because it is simply the most absurd example that anyone has ever sent me, I really have to say. The claims that are made by direct-to-consumer marketing clinics span just a really wide range. This is probably a more common one. A colleague sent this to me. It's from they were on a flight last summer, June 2019, on Alaska Airlines, and this was an ad that was in the magazine. Could stem cell therapy change your life? Do you have any of these 19 things? And then if you very carefully read at the bottom, these are stem cell therapies that have not been approved by the US Food and Drug Administration, the FDA. You're encouraged to consult with your primary care physician, blah, blah, blah. The only reason it actually says this is because California passed a law and said direct-to-consumer marketing clinics, you have to give a disclaimer of some kind. Up until last year, that wasn't even the case in reality. And the reason we have this proliferation of direct-to-consumer marketing clinics is the FDA has really hesitated to step in and regulate. Why? Because these are almost always either autologous, so for example, extracting fats from one person and putting it back into that same person, or cells that are largely, I'm going to come to this in a second, what are called mesenchymal in origin and thought to be kind of on the borderline of stem cells and what are called minimally manipulated, not changed very much in the lab, hypothetically meaning they have a greater safety profile. But I want to give you some examples of why you might want to think twice about that. So let's talk about this. What could possibly go wrong with the stem cell therapy? So in the first part of this talk, we discussed the complexity of stem cells and all of the things that we don't know in terms of developing stem cell therapeutics. And this is where what could possibly go wrong comes into play. We talked about the term pluripotent. So this is a cell that can make anything. If it can make anything, it can also make a tumor. And so if a cell that you transplant remains pluripotent and it continues to divide, well, that's something that could go wrong in terms of cell therapy. The cells that get transplanted or injected, they could get the wrong information. They could interpret signals from their environment, which is very complex, as we talked about. They could go to the wrong place. They could mature. They could specify. They could differentiate in the wrong spot, and that could create problems, adding to problems rather than making them better. The cell product could contain a contaminant that produces what we call an adverse reaction, a negative event. So in fact, in fall of 2018, there were 2 local production sites, Southern California production companies for minimally manipulated mesenchymal cells. I'll come back to mesenchymal again in a moment, that did just that. They introduced contaminants, and they had a host of adverse events. The FDA, in fact, came in and shut, for the first time ever, those 2 companies down and stopped them from distributing their minimally manipulated products to these direct-to-consumer clinics for that reason. And among the problems that they had was introducing bacteria into their product, which was then injected into people who ended up with very serious systemic infections as a result. What else could go wrong? Patients who are participating in this direct-to-consumer market could be excluded from future legitimate FDA-authorized clinical trials. So if, for example, you have a neurodegenerative disease, Huntington's disease, let's say, Parkinson's disease, ALS, and in some cases, there's a tremendous concern to move forward to having any kind of therapy as soon as possible. We actually have clinical trials coming down the pipeline for those particular conditions. If you've enrolled in something or participated in a direct-to-consumer marketing therapy, one of the exclusion criteria for those clinical trials is probably going to be whether you did something like this. So you take yourself out of the running. Patients may be excluded from future clinical trials. Some of these in the community here are between $5,000 and $20,000 a shot. And while it can be the case that patients and their families can be fairly desperate to try anything, it could be the case that that $20,000 could be better spent elsewhere. And last of all, something that I worry about a lot as director of a stem cell research center is a loss of public trust in science, right? So I'll give you an example. We run, as I'll allude to at the end, a community lecture series at UCI once a month. We invite people from the community in. I'll tell you more about it. I hope I see some of you there in the future. And we talk about different sorts of diseases and indications, the basic science of stem cell biology, and where we are on the clinical side, paired together. And in September last year, at our kickoff lecture, I had a lovely lady come up to me at the end. She waited a long time for people to clear and be done with their questions. And she came and she said, you know, I have a problem with my knee. And my friend suggested that I go to this clinic that's around the corner and get a stem cell injection. So I did. And then my knee swelled up. Should it swell up like that? And I had to go to the hospital, and I had to be on IV antibiotics, and it was a terrible experience. And we talked about, in fact, introduction of contaminants and these 2 local production sites that had been shut down. I was pretty sure that based on the timing, she had received a cell product from one of those places that the FDA had shut down. And she tells me this long story. I give her all of the cautions that we just talked about in terms of thinking about direct-to-consumer marketing clinics. And she said, yeah, but I understand what you do here. That's academia. But my question for you is, they're calling me. They would like me to go back and pay another $5,000 and have another injection. Should I do it? So we worry a lot about this loss of public trust, right? Because for her, it seemed completely reasonable to have confidence in what that clinic was marketing to her, as opposed to what I was trying to say ought to be things that we look at with caution. All right, so now I just want to give you a couple of examples as a slice. These are easy to find online. There are unfortunately a large number of examples that are out there online of things that have been the consequence of either direct-to-consumer marketing clinics within the United States or outside the United States. So this is a story that actually goes all the way back to 2012. I like to show it out here because this is a direct-to-consumer marketing clinic, one of the first in California, in Los Angeles. And they took a minimally manipulated product with the goal of, reducing our age-related lines under our eyes. And the physician that was involved said, it makes perfect sense to me that I would just give that in a single injection with the collagen that I normally give, a plastic surgeon who did this, right? So I'm just going to mix them together, and I'm going to give them to people at the same time. And then I don't have to do 2 injections, right? He's not a stem cell biologist. I don't think he understood the cells that he was working with, the product that he had received. He put those 2 things together, and that made an environment for the cells that said, oh, you shouldn't make more collagen, you should make bone. And in fact, if you put those 2 things together in a dish and you had tested it out, you would have stimulated those mesenchymal cells to make one of the multipotent things they can make, which are osteoclasts, which make bone, and he would have seen that. But that wasn't his game. He was just trying to be efficient in terms of billing within his clinical practice. And so this is an example of someone who ended up with bone growing underneath her eyes where all the injections had been made, and she had to have multiple surgeries to try and remove those bone pieces. It was very painful, apparently. So that's one example. This is an example that I highlight because it's something that I see a lot in the area of spinal cord injury. Again, my main bit of research. This is a gentleman who went outside the United States. In fact, he went to clinics in, I just want to read it off here, Mexico, China, and Argentina. He happened to have had a stroke that affected his spinal cord. He received stem cell injections from all of those 3 different places. Who knows what he got where, but at some point he got a cell that didn't listen to where it was put, and it kept on dividing, and it made a tumor. And so now, instead of being better, he's worse. He's gained significant paralysis instead of significant functional recovery. There are many examples that are along these lines. So these are the sorts of things to be aware of. So the cells that are marketed via these kinds of direct-to-consumer clinics are mostly this term that I've been throwing around, mesenchymal cells. So they're from one of those branches of the tree that we showed you at the very beginning. And they normally make or contribute to adipose tissue, bone marrow. You can get them from amnion, from placenta, all of these different sorts of areas. And so this is how direct-to-consumer clinics do the minimally manipulated. Usually it's bone marrow or lipoaspirate. It can be amniotic cells, which you'll see sometimes advertised at this clinic. But the conditions that these cells are marketed for are not things that are generally amenable to what we would think about therapy from a mesenchymal cell population. So this right here is one of the things that you should look at as an absolute red flag in a direct-to-consumer marketing clinic. Just like that Alaska Airlines ad, if the ad is for 50 treatments for 50 possible things using one cell product, that is the very definition of snake oil, right? So think about that before you engage. So here is a list of, from the marketed conditions that would be treated by these mesenchymal cells potentially. Everything from orthopedic conditions, knee conditions, pain, sports, neuroindications like ALS, Parkinson's disease, Alzheimer's disease, multiple sclerosis, cardiopulmonary disease, urologic conditions. You get the sense. All of these conditions, though, are not things that we would expect to be treatable by a mesenchymal stem cell. Mesenchymal stem cells have no business in anything to do with the brain or spinal cord, for example. So everything that has a red star here, we would never in a million years predict would be treatable by one of these cells. It is like asking a fish to ride a bicycle. So that's something to be aware of as well. There isn't one product that is going to be the magic bullet here. So how do we separate this hope that we have about stem cell research from the hype that I'm explaining to you in terms of direct-to-consumer marketing? Again, I'd like to share with you a couple of resources to think about. This is from an organization called the International Society for Stem Cell Research. It is the largest professional organization for stem cell biology in the world. And they have a fantastic website that is very useful. So it's called A Closer Look at Stem Cells. You can Google that phrase and you will find this website. And if you drill into it, they have some really nice patient resources. 9 things to know about stem cell treatments. And in fact, a list of patient resources. I think the most useful thing that they've developed is this ISSCR patient handbook on stem cell therapies. And so if you have questions that fall into this domain we've been talking about, I really encourage you to make use of this particular site. And actually, it occurs to me that I forgot something, so I'm going to back up one step. I apologize, and go here, because I'm going to show you at the end on our website at the Stem Cell Research Center at UCI. I mentioned we do this public lecture series, and we have one specifically that deals with this issue, hope versus hype, and how a clinical trial is different from what's happening at these regional places that call themselves clinics that are administering therapies. Sorry, now we got to jump forward. All right, so what's your checklist? Product is not a universal treatment. If a product claims that it can treat everything, that is not something that you want to engage with through one of these clinics. Either through the ISSCR or through this expanded lecture that we have online with us, it's really important to understand the difference between FDA authorized and approved clinical trials and what's happening in direct-to-consumer marketing. You should be confirming that there is FDA authorization for any of these regional clinics that you've been thinking about. I pretty much guarantee you that a stem cell clinical trial is only going on at an academic medical center. And so if it's a clinic that's in a strip mall, it's not a real thing. You can check online public resources include what's called PubMed. This is a scientific research database, but actually Dr. Google can be surprisingly helpful in terms of finding what preclinical data support. And by all means, you should always go back and talk to your clinician or specialist before you engage in something down this path. They're professional and it would be good to get their opinion. If you are asked to pay for a treatment, it is not a clinical trial. Clinical trials in the United States do not require you to pay. So if it's a $5,000 or $10,000 or $20,000 for an injection and they say the words clinical trial, it's not a clinical trial. Okay, so where does this leave us on the road to regenerative medicine and stem cell treatments? Now, I think it's really important to turn and talk a little bit about California Institute for Regenerative Medicine and the things that are specific to California in terms of stem cell research. So I mentioned at the beginning that we're the Sue and Bill Gross Stem Cell Research Center. We are a CIRM institute. What is CIRM? Again, California Institute for Regenerative Medicine. This was, as I mentioned before, created in 2004 when voters in California voted by about a 60% margin to support Proposition 71, which allocated $3 billion for stem cell research at a time when one couldn't do stem cell research otherwise within the United States. CIRM funds stem cell research at institutions and companies throughout California, and the express goal in their charter is to accelerate the development of treatments. So have they been successful in that? I want to show you a couple of slides of information about that. In fact, to date, there are over 60 CIRM-funded, FDA-authorized clinical trials that are ongoing in California or beyond that are using stem cells as a potential therapeutic or targeting a stem cell as a potential therapeutic. And in fact, if we include the number of clinical trials that are not paid for by CIRM but sponsored by companies, this is even larger. And it is directly due to that Prop 71 funding. So an important aspect of that is something else I want to introduce to you, which is an alpha stem cell clinic. And that is critical because it really is a huge distinction between direct-to-consumer marketing that we've just been talking about and how to think about potential and where things will go for stem cell therapies moving forward in a responsible, well-controlled, ethical clinical design kind of setting. And the Alpha Stem Cell Clinic is meant to be the answer to that. This is a place where a cell therapy clinical infrastructure has been created with CIRM funding, with scientific and technical and medical expertise to foster clinical trials, to evaluate and establish safe and effective therapies, and to develop and maintain the delivery of therapies that are specifically approved and authorized by the FDA, right, within a traditional clinical trial structure. There are actually 6 of them that are distributed across the state. UCI is one of them. So within our Stem Cell Research Center on the first floor, we have some clinical space. Dr. Daniela Boda, who's the head of neuro-oncology, is my clinical director for our trials. And in fact, if we look across the state at the types of indications that are going forward for stem cell research, which you can see on the right side of the slide here, these span ALS, I'll just read them off to you because I think you won't be able to see, brain injury and stroke, cancer, type 1 diabetes, eye and vision diseases, heart failure, hemophilia, HIV and AIDS, kidney failure, a number of neurological conditions, everything from spinal cord injury to stroke, severe combined immunodeficiency, so some genetic diseases, X-linked chromosome diseases, acute myeloid leukemia, you get the sense. So this is our particular clinical trial pipeline at UCI. We actually have 9 trials that are enrolling across a different array of diseases and conditions, including glioblastoma and 2 current diabetes trials. We have 12 that are in our current pipeline. And so what sorts of things then have come from stem cell research, not just at UCI, but across the state in terms of the CIRM network, and in particular, the Alpha Stem Cell Clinic, the ASCC network. So I want to give you one particular example of where I didn't imagine stem cell biology would take us when I first started down this path. Mind you, work from my laboratory and Brian Cummings' lab at UCI, we were one of these clinical trials. We did the preclinical data for 2 clinical trials that went forward with stem cells as a tested therapeutic for spinal cord injury. Brian is currently developing what he hopes is going to be a potential treatment to be tested for traumatic brain injury in the realm of neurological diseases. So we are intimately familiar with this process. What I didn't think about as we were thinking about cells for transplantation were the types of genetic diseases that are devastating that can also be targeted by targeting a stem cell as opposed to transplanting a stem cell. So you're all certainly old enough. You may not have been watching the same shows that I was. This was on in the 70s, and I remember watching it after school with John Travolta because it was very exciting back then. The Boy in the Plastic Bubble. And the idea here is that this was a kid who had been born who couldn't be exposed to the outside world, right? So he had what's called severe combined immunodeficiency, meaning he failed to develop a normal immune system. It was very exciting because it was John Travolta, but it actually was real. And so this is one of the patients that was characterized at that time. So this is a parallel article. This kid was also in the 70s. The amazing thing is that it's a hematopoietic stem cell that has gone awry. It's a genetic loss, a genetic mutation, and you can fix it by replacing the hematopoietic stem cells. And so this is a little girl, Evangelina Vaccaro, who's amazing. And she was the first patient who was cured of severe combined immunodeficiency through the Alpha Stem Cell Clinic Network by a gentleman who does research in this area, Don Cohn at UCLA. In fact, at this point, there's over 40 severe combined immunodeficiency kids who have been treated from across the country at this point. Most of them are living normal lives right now. So to give you a couple of other examples, now coming back closer to UCI, in fact, one of our clinical trials ongoing right now is for a retinal disease. It's called retinitis pigmentosa. That's Henry Claassen at UCI. A parallel one of our trials is targeting glioblastoma stem cells. And this is Daniela Boda, who I mentioned is my clinical director, that's showing some really, really promising work just within our own particular pipeline. So is all of this worth it then? So on one hand, I find that really compelling and motivating in my own research, that in fact, we're at a point which when I was in graduate school, I never dreamed that we would be, where we can think about moving things out of the lab and into clinical trial. And that was, I think, the crowning experience of my academic career, was going to clinical trial with the cell therapy that we had developed in 2010. Is it worth it on other scales, though, I think is a legitimate question to ask. And so I'm going to just point you to this example. So this is a economic policy, economic study that was conducted last fall by University of Southern California. And again, this is easily findable online. Just looking at what the economic impacts of that $3 billion investment by Californians were for stem cell research. And it was a little surprising. I think it would have been great if we had broken even, but in reality, what their calculations are is almost $11 billion of additional sales revenue to the state from that $3 billion investment, 640 million or so of additional state and local tax revenues, 726 million of additional federal tax revenues, and over 55,000 new high-tech jobs that were created as a result of that investment. Hype is easy. How does hope get started then? And here's partly where I want to make that point. And like most of what happens in the world, it starts with a pipeline, and that pipeline can mean different things when it comes to different sorts of areas. Here, in terms of stem cell biology, we talk about a translational pipeline. And so, so this says, if anybody can't read it, it's nothing a few stem cells and another 75 years of research can't fix, and it definitely feels that way sometime. But again, I just want to point out, stem cell science has moved really, really fast. My lab did its first stem cell study in 2002. We were in clinical trial by 2010. So 8 years is a pretty quick move through that clinical translation pipeline. I think one of the things that's important to understand about that is that research is traditionally supported by the federal government. This is actually the reason that the National Institutes of Health exists. It's the reason that not a lot of patents are filed under National Institutes of Health funded research, because the goal is to move that out into the biotech sector to be able to build the economy and to be able to move things into actual application for real people. The problem is that that funding for research has steadily fallen, and we're actually seeing the consequences of that a little more indirectly right now with the coronavirus outbreak. So I would be glad to talk to you my particular opinions about that at the end if anyone's interested. But the idea here is that at our peak of federal funding for research, no matter how we measure it as a percentage of gross domestic product or total dollars, we've fallen to about 1/3 of our peak and a half of what our funding was at the start of the war on cancer when we correct in terms of cost of living increases, right? So we are not funding research at a robust level in this country right now. So things like support from the California Institute for Regenerative Medicine are actually really pivotal and quite important. And in that regard, new drugs and inventions come from academic research institutions. And this study I like is a great example of this. So this is a PNAS paper that looked at the contribution of NIH funding to new drug approvals, so what are called new molecular entities, or NMEs, over a 5-year period. And they just asked for NIH-supported research, we would assume a parallel for CIRM-supported research, how many new drugs that we made actually got their start with federal or state funding for research. So there were 210 new molecular entities approved within this 5-year period. This is the participatory part of the lecture. Who wants to take a guess at how many of those could be traced to state and federal funding? Half, 25%, 75%, 80%? Do I have any takers on another guess? 10%? Yeah, 100%. So if you don't drive the engine of research on this translational pipeline at the bench by funding, you don't get anything out to the other side. And I think that is a really important lesson to take away. If that is the one thing that I leave you with today, I hope it's that. So how do we do that at the Stem Cell Center? We try and make people more efficient. So the reality is that if we're at 50% of what funding for research is, we have actually more investigators doing research now than we did 40 years ago, certainly than we did in the 1970s. It's really competitive to get those grants. So we work hard to try and make our people more competitive for that. We do that by a seed grant funding program. We've invested about $450,000 in people over a 5-year period that we have quantified so far. From that, we have about $11 million in new grants that have come into UCI. So that's a 25-fold return on investment. And those include things like research from Matt Blurton-Jones, who's developing stem cells and a new model of Alzheimer's disease, thinking, again, along the lines of therapeutics for neurological conditions. Dr. Leslie Thompson just got a huge CIRM grant. She's advancing through, in a clinical stage grant now, to a stem cell therapy as a clinical trial for Huntington's disease. Brian Cummings, I mentioned earlier, has similarly a large CIRM award right now that these are all things that came directly out of one of our seed grants, advancing towards a cell therapeutic for traumatic brain injury, which is a huge but silent epidemic across the country. So how can you help? Well, you can participate. I think one of the things that we're very concerned about this year is that we will have a ballot initiative, again, for funding for California Institute of Regenerative Medicine, that we hope will be up on the ballot this fall. We're actually quite concerned right now because of the coronavirus outbreak that it's going to be difficult to educate the community about, stem cell research and kind of where things are. We need 1 million signatures. I should say Americans for Cures, who's sponsoring the initiative, needs 1 million signatures by the end of April in order to get it on the ballot initiative. So I would encourage you, if you're willing to educate yourself about that, AmericansForCures.org is a great place to do that. And consider that ballot initiative if hopefully you see it in the fall. Contact your state and federal representatives and indicate your support for stem cell research if you agree with me. You can sign up for our newsletter. You can follow us on Facebook. Judy Beck is at the back and had some information for us. You can certainly visit our website and get information. And UCI is in the middle of a huge capital campaign right now where we're trying to offset this loss of federal funding. So we certainly are very grateful for gifts from the community. And I just want to highlight that while research, I hope I've shown you, drives the US economy, in reality, less than 4% of annual giving in the United States goes to support scientific research. So maybe something to think about. I want to point you to 2 other resources before I stop. One is this set of whiteboard videos. This is at the AmericansForCures.org site. And it's a really great set of simple snapshot tutorials. They're all like 5 minutes long, just to explain the basic biology of stem cells, how stem cells might be used to treat retinal diseases, right? Different areas. They have them that span a wide range of the spectrum of diseases that most people are interested in, and it's a great resource to get information. We also, because of the community lecture series that we're doing, we record those. Those are up and on our website. So I already highlighted Hope versus Hype as one example of a video that hopefully could be of some use in terms of the community. And, but we have a lot, and we have a YouTube channel that spans all of these. This is Matt Blurton-Jones, who I was just referencing, and someone asked about Alzheimer's disease. He has one about Alzheimer's disease with one of our clinicians. And then we have this community lecture series. Unfortunately, we know already, because UCI just shifted its policy yesterday, that we're going to have to cancel, well, which one am I at? Insights and, yeah, we're going to have to cancel the March 24th one. We're going to do it online instead and do a rollout for that, but we're not going to be able to have a gathering. We normally have about 160 or 170 people at these, and so right now we're trying to limit anything that's over 100 in terms of people gathered together. So that will be online. We may be back in business for April, but if not, that will be online too. We're figuring it out right now. And that's where I'm going to end.